Start recording, share the screen. Let me see, is it download? It might not be if I don't remember where. There it is, Zoom. So there's our lecture meeting, and it's not showing me. Here's the lab meeting, and it's these two files until you end the meeting, and then these two files are converted into this file, and this is the file which you can play the meeting over YouTube or something like that. These files, I don't know what they are. They're something specific for Zoom, and at the moment they're recording the information in these two files, and when you end the meeting, these two files will automatically be converted into the Zoom file. And I'll change the name of this and call it the lecture file. All right, any questions about any of that? I don't have my syllabus up anymore. Let me see if I can get it back. I shut it down. But are we recording this? Let me see. Yes, we are. All right. There we go. So if you remember, I said we would spend a little bit of time in the lab time spending on the lecture. And in fact, if you notice this link for Zoom, I've got lecture and lab. So we'll spend about 15 minutes on the lecture before we start the lab. And the reason is, for some reason, whoever did the summer schedule, they kept the lecture time the same as the 10-week class, and then they increased the lab time. And I don't really need the lab time increased. I need the lecture time increased. And I don't know how much time it should be, but this is the same number of hours for 10 weeks, the same time, meaning an hour and 20 minutes. For 10 weeks is what we normally have. And we only have 8 weeks, so you'll only have an hour and 20 minutes for 8 weeks. And I need a little more time to cover the material. Any questions about any of that? All right, so after I've spent about 15 minutes on the lecture, which will be chapter 1, we'll then talk about Lab 00, Laboratory Safety Training. And you will turn in a lab worksheet for Lab 00. Any question about that? The lab worksheet for Lab 00 and Lab 01 is only worth 2.5 points. So you need 2 of them to make a full lab. But other than that, they do count as a lab. All right, so let's return to the lesson in the lecture. So I talked about microbes being important on the planet Earth in producing oxygen, as well as helping to recycle nitrogen in the Earth's atmosphere. Any question about any of that? Well, microbes are also important on the planet in recycling other nutrients. For example, this is a sewage treatment plant, and humans are responsible for making the water waste by flushing their toilets and flushing things down their drain. And humans break the waste down. That's why we built these facilities, right? Anyone object to that? That humans break down the waste in these facilities? Nobody's objecting? Well, somebody should, because humans don't break down the waste. We bring it to these facilities, and then we allow microorganisms to grow in our waste and break it down. And most of that waste will be broken down into carbon dioxide and water, like we do when we're eating food. Well, the microbes, they take the toilet paper and the other stuff from the toilet and break it down into mostly CO2 and water. And the point is that the sewage treatment plant, it's the microbes which are breaking down the waste, not the people. And we have microbes breaking down other waste, like when a tree dies in the forest, it's largely microbes that break down the tree and recycle the nutrients. And why the nutrients recycle and can be used by other organisms is because it's broken down by the microbes in the tree and in the toilet waste. And then other organisms can use those nutrients. Obviously, the organism like a green plant can use CO2, but when you flush your toilet and you're eating, I don't know, something like the chromium in your diet that is not consumed and flushed down the toilet, that will be broken down by the microbes. And then other organisms can use that chromium. And that's true for all other nutrients like calcium and phosphorus, nitrogen and carbon too. And we'll talk about that. The carbon in the toilet waste will largely be converted into CO2, and then green plants and microbes can take in that carbon dioxide and then recycle it, making it into glucose, which then can be eaten by an animal or a person. And green plants can make other molecules too besides glucose, but we're just going to stop at glucose. Okay, everyone understand how important microbes are for breaking down the waste in a sewage treatment plant, as well as like a tree that dies in the forest. Any question about that? If not, let's move on to what microorganisms are. We can use that term synonymously with a microbe. These are organisms which are too small to be seen with the unaided eye. You need a microscope to see them. They are single-celled organisms that come in many shapes and sizes. It includes bacteria, archaea, yeast, molds, protozoans, some algae. If the algae is microscopic or single-celled, it's a microorganism. If the algae is something that washes up on the ocean beaches, and we usually call that kelp, and you can pick it up and swing it, that is not a microscopic organism. And then there's viruses and prions, which are also considered microbes. And you can call them a microorganism. But if you call them a microorganism, you should realize that these organisms do not have cells. So they are special in that they do not have cells, and well, you could argue they're not organisms because they're not cellular, so they're just special. All of the other organisms that we talked about have cells, and they are, at least the ones that we're listing here, are microorganisms. Okay? Viruses and prions are microbes, and they are special because they do not have cells. Any question about any of that? Microorganisms are almost everywhere. So we can call them the ubiquity of microorganisms. Almost any place you look on the planet Earth, assuming you're looking, someplace around the surface, you can find microorganisms almost everywhere. They play beneficial roles in numerous processes, and I've spoken about 3 of them. Recycling of nutrients like nitrogen in the planet, the production of oxygen in the planet, and then the recycling of nutrients from dead materials and waste materials. So they play beneficial roles in numerous processes. And they're numerically the most abundant life forms on the planet Earth. You can find more microbes than any other form of life. Now, I've talked about cells. Let's talk about what is a cell. A cell is the basic structural and functional unit of all living organisms. But wait a minute. Didn't we just call viruses and prions a microbe? And they don't have cells? Well, yes, that is correct. When I say that cells are the functional unit of all living organisms, it's debatable whether viruses are alive or not, and we're not going to get into that debate. They certainly don't have cells, and prions, everyone agree, would say that they are not alive. Okay? Cells are a highly organized compartment. They're bounded by a thin, flexible structure that we call the plasma membrane or the cell membrane. And all cells contain 3 components. They contain the plasma membrane, obviously. They contain the cytoplasm, and that's the liquid portion inside the cell other than the nucleus, and prokaryotes don't have a nucleus. We'll talk about that later. And then cells also have a genome, which is a DNA molecule. So those are the 3 components that all cells have: DNA, cytoplasm, and a plasma or cell membrane. And you'll note we're using the terms plasma membrane and cell membrane interchangeably. And I guess I can say we use microorganism and microbe interchangeably as well. If we look on this picture, in every centimeter, can you find a microorganism in every centimeter of this picture? Like on the plants, in the rock, or the soil, whatever that is, in the water? Can you find a microorganism everywhere in this picture?
Yes.
Yes, you can. How about in this picture? When we look in the air, when we look on the surface of the water, and we look underneath the surface of the water, in every centimeter, would we find a microorganism?
Yes.
I didn't hear that answer. Somebody answered. That's a yes.
Yes.
Is anyone going to guess? Come on, people.
Yes.
Oh, skip this one.
Yes.
And huh? Sorry, I didn't hear that. All right, we'll skip this one and come on to this one. Here we see the air once again, and then the plants, and then the top of the soil, and in the soil, and then deep down into the soil. Well, we're going to find a microbe in every centimeter. A centimeter, by the way, there's 2.4 centimeters in an inch, if you don't know that. So it's smaller than an inch. Will you find a microorganism everywhere in the soil, on the surface?
Yes, you will.
You will. Good. You will. Obviously, you find it on the plants and on the top, and you do find it in the soil. And I don't expect you to know this now, but you can find microorganisms down about 1 mile from the surface. And that's actually true for the surface of the land and the bottom of the ocean, which is the surface, I guess, of the land underneath the ocean. You can look under 1 mile of the ocean floor and find microorganisms. And up in the air, you can find microorganisms floating around on the gusts of wind up about 1 mile in the air. Okay? So you can find microorganisms in a wide variety from the surface. And you obviously find microorganisms everywhere on the cat and the person, every centimeter. How about in the desert? There's certainly less life in the desert. Would you find a microbe in every inch of the desert sand, or I should say every centimeter of the desert sand?
Yes.
Yes, you would. And let me tell you what this is. This is a volcanic vent. That red stuff there is lava. And this is the vent's crater. And I don't know what this is, rock around the vent. Would you find microorganisms everywhere in this picture?
Yes.
Somebody said yes. You do find it in every place in this picture except in the lava. This red stuff is lava, and that's way too hot for a microorganism to survive. And so if a microorganism were to fall on the lava, it would die, it would be killed. Okay? So, and everywhere in this picture, we have microorganisms, and everywhere on the planet, in every inch, except in the lava. All right? Where is everywhere? Where do we find microorganisms? Every environment where the land and the water is, in the atmosphere, as I stated, about 1 mile up, subsurface on the land and the ocean, about 1 mile down, on every plant and animals. Essentially everywhere liquid water is present. Life can live between a minus 20 degrees Celsius, and remember 0 degrees Celsius is freezing, to 120 degrees Celsius. And remember 100 degrees is where water boils. So life can live at temperatures below freezing and above boiling water. All right? And this will be the temperature Fahrenheit, at minus 4 degrees and 250 degrees Fahrenheit. At lower temperatures, the microbe would freeze, so it's not metabolically active, but it's not necessarily killed. Okay? And it depends on the microorganism whether it would die when it's frozen. Most bacteria, when you freeze them and then thaw them out, about 50% of the cells will still be alive. And that means they're resisting freezing, or at least they're resisting dying from freezing. And there are some microorganisms, we'll talk about them later in the term, that can live at temperatures above boiling water. Okay? All right, any question about any of that? If not, we're going to stop the lecture here, and I'll start the lab now. All right, so the lab is Lab 0, Lab Safety Training. Get there. Do I have the lab in a different place, or oh, it's here? Sorry. So when you work on the lab, you will be reading the lab module, which, let me show you that. Come on. And I'll go over the lab module with you. But you'll be reading that, and you will not be answering the questions in the lab module. There's the lab. No, it isn't. Where's the lab? There it is. So this is the lab module 00, lab safety. And you have a worksheet for lab 00. And let me go to that worksheet. And there's the worksheet. So download the worksheet, answer the questions on the worksheet, and only upload the worksheet. And I'm an instructor, so I have the grader. I don't have a link for downloading a lab worksheet, but you won't have the grader sheet and the download submissions. You'll have an upload submission button or something like that. And I can go to student view and see that, what you'll have. Start assignment. And there's the submit assignment. So download that and fill in the answers. You can type it. I've had a few students who actually print it out and then write it in by hand, and they must have a scanner, and then they submit it as a scanned assignment. That's okay too. It's probably easiest just to download it, type in your answers if you have Microsoft Word, and then submit it. If you don't have Microsoft Word and you have another program, understand that it has to be submitted at a certain file type, and I generally accept Microsoft Word documents, the modern ones or the older ones, the .doc. I also accept rich text formatted files, and most word processors, actually I think all word processors will save it as a rich text format, and I usually will allow a PDF document. Sometimes I'll allow you to submit a text document or a HTML document, and there's one other one, a PDF file maybe, whatever, okay? But I always allow the Word document or the doc, the DOC and the DOX, a rich text formatted document, and I always allow a PDF file also, okay? So if you don't have Microsoft Word and you use another program, just make sure it's submitted. Oh, there it is right there, the file types. So with this one, I'll accept doc, docx, rich text format, a text file, a PDF file, and an HTML file. All right, why is that fighting me? Let's go to the lab module and go through it. I think that's it, but it might be that one. So let's open both of them just to make sure. Yeah, it's the second one. All right. Let's see, I'll put that at the very top, see if it goes there. There we go. Laboratory module 00. Laboratory safety. So online students will not be working in the lab, but you need to know this material as many of the rules will be relevant during your careers. For the learning objectives, you need to be able to distinguish between safe and unsafe laboratory practices, understand where relevant laboratory safety equipment is located in the lab, understand how to safely dispose of contaminated materials. And we're talking about materials contaminated with microorganisms. I was hoping that I had this highlighted, but it looks like I do not. For cover. Here it is here. Microorganisms are too small to be seen with the naked eye. Viruses and prions are included. And there is one other organism that is included as a microorganism and studied by microbiologists. These are helminths, which are more commonly known as worms. Okay? And we'll talk about why we study them in the lecture. I'm not going to talk about that now for this lab, but the adults are clearly not microscopic for most of the worms, but their infective stages are microscopic. And then they can give disease. So microbiologists study all organisms that cause an infectious disease. The phrase ubiquity of microorganisms refers to the concept that microorganisms are essentially everywhere. As we showed you on the picture of the planet, microorganisms were everywhere we looked except on lava in the volcanic vent. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn basic lab protocols, including the use of solid media, labeling lab cultures, procedures for disinfecting the lab benches, and procedures for washing our hands. When you come into a lab, you go through a specific number of steps for what you do before you begin work in the lab. And the very first one, I guess after you walk in the door and put your things in the cubby hole, the first thing you do is you go and wash your hands. We work with culture media. A culture media is also called the growth medium. It is a sterile laboratory preparation that contains a variety of nutrients that are required for microorganisms to grow. Culture media in the lab may be liquid, and these are called broths. So a broth solution, or the broths, and are commonly used in test tubes. We can also add agar to the liquid medium to result in a solid medium. An agar can be used in a test tube as well as in an agar plate, meaning you pour the agar while it's hot into a plate. Agar is a substance derived from seaweed, and we'll talk a little bit about that later. We have a video for you to watch on preparing pour plates. So you should note how the culture medium needs to be sterilized before use. And then you pour it. Here is showing you glass test tubes. This one is a liquid media, and you can kind of tell because it's clear or very transparent. And the agar tubes, although that one is translucent, you definitely can't clearly see through it. And the agar in solid media can be in a deep, that's this tube here, where you just pour the agar in and it solidifies on the top. You can also pour the agar in the tube and then put the tube at an angle, and you can get a slant. We call that a slant. And then you can pour the agar in a petri dish, or we call it a plate, and then we have agar on a plate. And so here's different media with agar plates. We're going to be talking about different types of media, and the first type we're going to talk about is general purpose media. And this lab will only discuss general purpose media. These are non-selective media, and they're usually complex in composition, and they support a growth of many different organisms, but not all organisms. Fastidious organisms will not grow on a general purpose media, so we grow non-fastidious organisms on the general purpose media. And they use a variety of peptone, yeast, and beef extracts to supply nutrients to the cells. What did I want to talk about there? One purpose for growing general purpose media is simply that you can grow many organisms like E. coli, Staphylococcus epidermidis on them, and it's very cheap. So the general purpose media is used a lot because it's the cheapest media. There's different general purpose media. Let's talk about this one first, number 3. Nutrient agar, because it's the simplest. Tryptic soy agar, it uses proteins from soybeans to make tryptic soy agar. And then this one is nutrient agar, the same as number 3, but we enrich it by adding yeast extract to the media. Okay? So it grows a variety of many different organisms, but it does not grow all organisms. And anything that's fastidious, something that requires certain components to be added before it can grow, will not grow on a general purpose media. An example of a fastidious organism is Streptococcus pyogenes, which causes strep throat. It will not grow on a general purpose media. Okay? Any question about any of that? All right. In the lab, we need to label everything, and it's vital that the Petri plate or the tube is labeled correctly. You always label the Petri plate or the tube with 5 possible things. You put, or 5 required things. You add your name, and you may use your initials. You add the date, the type of media it is. You can add the incubation temperature, but I tell my students that the temperature is optional. And then you put the inoculum, what you're growing. If you know what you're growing, you write it. Like this is Travis, and there's the date, right there. And this is the media, TSA, and he's got the temperature that he grew it at, 37 degrees. And then this is what he grew, E. coli. If you don't know what you're growing, you then say something descriptive about what it is. Like if you're growing organisms you got from the air, you would write something like air or air culture. But all the others are pretty straightforward. Any question about that? So on the plates, you always write on the perimeter of the bottom of the plate, as shown here. And why you don't write in this region of the bottom of the plate, because if you did, you might harm your ability to see the organisms growing on the plate. Like if we had a black colony growing right there, and you were to write Travis right there, you might hide the black colony. You always write on the bottom of the Petri dish, because if the lid gets separated from the bottom, the organism is always going to be on the bottom, because that's where the agar is. And I once had a co-worker, and she had a whole bunch of Petri plates, and she was carrying them around on a tray, not exactly a box like this, but a tray, and she tripped, and she was carrying over 300 Petri dishes, and all the lids got separated from the plate bottoms. And guess what? She had written everything for that experiment on the lids, and nobody knew what lids went with which bottom, so the whole experiment had to be thrown away. So we always write on the bottom, so that if we find the plate separated from the lid, we will have what is on this plate, as well as who made the plate what date it was made, what the media is, and possibly, this is optional, what temperature it was growing at. Another reason for writing on the bottom of a Petri dish is, I don't know if you can see this, kind of, is that you will grow your plates in an incubator upside down. And so you can read the bottom of the plate because it's upside down. And that's just fortuitous, meaning it's not designed that way. We grow the plates upside down in the incubator because there's moisture in the agar. And if you grew the plate right side up, the moisture would condense on the top of the lid, and it would condense and eventually form a drop. And where is that drop going to drop? It's going to drop into the culture, and that will disperse the culture, which could ruin the culture. The water is likely to be sterile because the Petri dish, the lid should be sterile. Okay, although having the water drop down would increase the risk of contamination, but the water is likely to be sterile, so it doesn't contaminate the dish, but it can ruin the experiment because instead of having isolated colonies, you'll now have the water spreading the bacteria so it starts growing everywhere. Any question about any of that? So you always write on the bottom of the dish, and when you have your Petri dish and put them in the incubator, you always insert it into the incubator upside down. And that way water will not condense on the lid and drip down into the culture, ruining it. And if water did come out of the agar, which I suppose it does, it will condense on the agar, on the bottom of the lid, the bottom of the plate, and then the agar will wick the moisture around so there won't be any droplets dripping down. And even if it were to drip down, it'll drip into the lid, and there's no bacteria growing in the lid, so we don't really care if it drips into the lid. Okay? Any question about any of that? How you add the labeling information on a tube is you get a piece of tape, and then you write the information on the tape, and then you put the tape on the tube. So it's going around here. Generally, you don't want the tape to be like right here, because we'll be growing the organisms here. We might be growing it here. So you don't want to block what you're seeing. So you can either put the tape, like up here would be best, because there's no organisms growing here, or on the test tube, like right here, would be best. And then you don't block what you're seeing, or could see, in the tube. Any question about any of that? All right. The inoculum is what you place in the tube to grow. Usually, we will sterilize an inoculating loop and put it into a sterile culture that only has one organism in it. Take the organism out and transfer it into a sterile culture, and that is called inoculation. Incubation is the placement of the inoculated culture in an appropriate environment for growing it, which is usually an incubator. Already talked about this. All right, let's talk a little bit about colonies. Bacteria colonies tend to be smooth, textured surfaces, meaning smooth colonies that have smooth edges. There are exceptions, but most bacteria colonies are smooth. In comparison, most fungal colonies tend to be furry or hairy looking. So usually when you look at the colony, you can tell whether it is a bacterial colony or a fungal colony. You should also know that a colony is composed of tens to hundreds of millions of cells. So 10 million cells to 100 million cells. And they all may be derived from one cell or a few cells that were placed there during inoculation. And then that one cell or few cells grew to form the colony. And to seed the colony, it has to be at least 1 million cells. Okay? But some colonies can be 10 to 100 million cells in the colony. That one cell will simply divide into two cells into four cells into eight cells into 16, etc., until we get enough cells that we can see the colony. And these we call that bacteria grow by binary fission. We'll talk about binary fission later in the lecture. We might talk about it in the lab. Now here's a video for you to watch on binary fission. And here is a picture of where this person is incubating their cells, or their petri dishes. And incubators can be simple, where all they do is heat and add a little moisture, or they can be complex, as this one is, and it looks like you can change the gaseous components in this incubator. You might be able to change the humidity as well. I mentioned that bacteria colonies are smooth and have a distinct edge, like this colony has a smooth edge, and it's distinct. Whereas this one, it's kind of hairy or fuzzy, and the same with this one, which the black part is showing you it's more, I don't know, fuzzy than the white. But these two are fungal colonies, and these, as well as that one and that one, are bacterial colonies. Any question about any of that? You can look at the colonies and get different characteristics. You can note their shape, a circular colony, an irregular colony, a filamentous colony. You can look at the margin, that's the edge of the colony. And if the margin is circular and smooth, we call it an entire margin. And then there's undulate, lobate, curled, rhizoid, and filamentous. Whenever I ask you a question about what is the shape of the margin, I won't ask you to distinguish between undulate, lobate, and curled, and I won't ask you to distinguish between rhizoid and filamentous. But you should be able to distinguish this group from this group from that group. Any question about any of that? All right, you can look at the elevation of the colony. Some colonies are flat, some colonies are raised, some are convex, some are pulvinate, and I will never ask you, is this colony convex or pulvinate? But I could ask you, is the colony umbunate, or is it convex, or is it raised? To see the elevation, you usually have to take the lid off the petri dish and then look at the colony at an angle. Simply looking at the top or the bottom of the plate will not tell you the elevation. But you can look and see the shape, the margin, the size, just by looking from the top of the plate. The size, punctiform, that's about the size of a pencil. I don't know if I have a pencil here. Well, I got a pen, that'll be good enough. So the tip of the pen, that's punctiform. So it's a pinprick. Small is something smaller than the eraser on a pencil. I don't have one, but it'll be about this size. Okay? And then large is something that is larger than the eraser on a pencil. And moderate is something between large and small. You can look at the colony and say whether it's smooth or rough. So when we look at this picture here, we would say that colony is smooth. Glistening, that's whether it's shiny or not, and the one I showed you was shiny, or whether the colonies are dull. You can also say whether the colony is pigmented or non-pigmented. This white colony and that white colony is obviously non-pigmented. How about this colony? Is that pigmented or non-pigmented? Or I could say that colony. Are these pigmented or non-pigmented?
Non-pigmented.
Yeah, pigmented means the colony has color, so these yellowish ones would be pigmented. The white, the off-white, the gray, they're non-pigmented. Then you can look at the optical properties, and most colonies are opaque. They don't let light through. Some colonies are translucent. You can see through it, but it's not perfectly clear. It's like lightly muddy water you're looking through. And I don't see any translucent colonies in this picture. All of them are opaque. And then there's transparent colonies, and you can see right through a transparent colony. They're very rare, the transparent colonies. I have seen them, but only a few times in my life. Oh, here's a better picture. This colony right here would be translucent, and that is either the center of this colony where it's opaque, or it could be another colony growing on top of this colony. But this colony would be opaque, that colony would be opaque, that colony opaque, and that colony is translucent. Any question about any of that? Now the good news is, if I asked you about colony characteristics, I will provide you with this table. Okay? And then you have to tell me the colony characteristics. The only exception I can think of, you should know what is a pigmented colony and a non-pigmented colony. And I might not provide the table if I ask you that question, and this table won't really, I guess it would help you. It would help you on that one. But that might be one you should know. All right, here we're looking at different colonies. And when you talk about the abundance, that's simply the total number of colonies on the plate. And this plate has, I don't know, 30 to 50 colonies. And so we'd say the abundance is 30 to 50. And I'm not going to actually count them, but you could. I've got an easier one to do. And we can say the abundance of cats in this picture is 7. 1, 2, 3, 4, 5, 6, 7. So the abundance is simply the total number of colonies on a plate. The diversity is the number of different kinds of colonies on a plate. So the diversity of cats is 2. There's lions and there's cheetahs. Okay? So learn these two terms, abundance and diversity. And like I said, the abundance here is something like 30 to 50. I'm not going to spend the time to count them. And the diversity is, well, there's 1, there's definitely 2, probably 3, 4, and that might be different than that one, but it might be the same. But this one looks different, so that's 5. I don't think we have a small white colony, so that's 6. And this one has a different edge, so that's probably 7. And I don't know if that's the same or different, so we won't count that as another. Okay? So there's at least 7 different colonies for diversity here, and there could be others. Like that one looks a little different in color than these two, so that would be 8. There might be 10 or 12 for the diversity here. And when I'm asking you for diversity, I'm not going to be nitpicky that you have to get my number, which I think I told you there's 7 or 8 and someplace to 12, as long as you're near the range. But if you tell me 4, and I look at that and I say, well, that's different from that one, 1, 2, 3, and then that one's different too, so there's obviously more than 4 because all of these are off-white or white, and then there's 2 over here which are definitely colored, so there's clearly more than 4. So if you were to say your diversity is 4, I would mark it wrong. But if you said your diversity is close to my numbers, like 5 or 6, I'll probably let that go. And if you said something like 15 to, I don't know, 15 to 17, I would let that go too. But if you had too many, like you said 40, and there's at most 50 here, I would say that's too much diversity, because clearly this one, this one, this one, and this one are all the same, and then this one and that one are probably the same, and that one, this one, and that one are probably the same. Okay? Are you getting my drift? So you have to be close on your diversity. With your abundance, you also have to be close, because I'm not going to expect you to count all of the colonies here. Okay? Did I have another plate? This one is even a bit much, and it'd be really hard to count how many colonies you have in this region and this region. You might be able to count them there. But this one is a bit much, so I'd take an estimate here. Okay? But the abundance, if you can actually count them. And like I said, that's a bit much, and so I don't expect you to count that. But if it's 10 or less, I do expect you to have the correct abundance number. Okay? Like I expect you to know the abundance in this picture is of 7 cats, and the diversity is 2. Any question about that? All right, you should know the terms of this lab. They're listed here. And then you should go through the lab. You don't need to really do anything with the references. Oh, good. There's not even any questions on Lab Module 00. So to get the questions, you have to go to the worksheet. And there's the worksheet for Lab Module 00. Yours would be right there. And this link will have both of them. Do I still have that there? Yes, I think I do. No, I don't. We'll go there. It's going to have 2 links. You only need to do this one for today. And then on Thursday, we'll do this one. Okay? And then both of these are going to be due on Saturday. So you must work on the lab and submit it by 11:59 p.m. This Saturday for full credit. Any question about any of that? All right, let's look at the questions. Go through the questions, read through it, and this is actually hard to do. I didn't even understand what this was about because I don't have a printer, but I learned from students that if you print this page out, that something prints here, and I'm going to tell you to ignore the first page and go to the second page. I've got the questions here. So in the lab, what do you always wear for safety before you enter the lab? So this question is asking you, what do you have on before you open the door to enter the lab? Okay? So obviously, you get the lab goggles in the lab as well as the lab gloves. So I really should not accept that for Rule 1 because you don't have those on before you enter the lab. And there are 4 things you should answer because I've got 4 asterisks here. So the asterisks tell you how many answers I'm expecting. And then after you enter the lab, what do you put on to keep yourself safe in the lab? And I'm only asking for 2 things here, although I can think of 3 things off the top of my head. And these should not be the same as in Rule 1, although I will allow some overlap, okay? In Rule 3, you'll have to look online what a safety data sheet is, and each chemical has one. And what's on the safety data sheet? What does it describe? And give me 1, 2, 3, 4, 5 answers for what the safety data sheet describes. You should also know the location of your safety equipment, and that would be equipment you find in the lab to keep yourself safe. And that would be something like a fire extinguisher. So I'm giving you the name of one of the safety equipment. Okay? And obviously you're not going to actually go in the lab, so every lab, if you were to go into it, has this safety equipment. And I've never had to use a fire extinguisher, but it's there just in case a fire happens, especially if somebody catches on fire, and then we can put it out. Okay? And there's other safety equipment in the lab. You can read about it. It's in Lab Module 00. And then what can you take into the lab? So there's 2 things you need to answer for that. And actually there are some things to take into the lab. For example, as you notice, I'm wearing glasses. Do you think I can take those into the lab and then take them out of the lab? The answer is yes, we will allow that. Okay? You shouldn't get them contaminated with bacteria, but we will allow certain things into the lab. And then the third one is, can you eat and drink in the lab? And if you get that one wrong, I should fail you, but you should be able to answer that from Lab Module 00. And then list 1 item of laboratory attire you must wear in the microbiology lab. List 1 item that is allowed in the microbiology lab. And then list 1 item that's not allowed in the microbiology lab. Oh, and that's right. Sorry, I forgot. Lab Module 00 is due Friday at 8 a.m. of week 1. I forgot about that, so I've got to change the due date in Canvas. The reason is I need to take attendance for people taking the class. And if somebody registered for the class and then doesn't show, I need to drop them on Friday. So you must submit Lab 00 by 8 a.m. on Friday so that I can determine who's taking the class and who is not taking the class. Boy, I'm going to have a hard time here because I've got both of them together. All right. And so if you do not submit the assignment by Friday at 8 a.m., I will drop you from the class. So please submit Lab 00 by Friday at 8 a.m. Any questions about any of that? If not, I will be here until 8:30 to answer any questions. Okay? And please try to answer your questions before 8:30. Don't come in at 8:29, because when the last student logs off, I will probably log off. So I will be here at least until 8 o'clock. But if everybody logs off at 8 o'clock, I will log off as well. However, if students continue working on the lab until 8:30, I promise I will be here until 8:30 to answer any questions. All right, so go ahead and get started on the lab. And I'm going to change the due date for Lab 00 in Canvas.